10,000 years ago, humans had not yet invented the wheel. We had no written language, no cities, no agriculture beyond its earliest experiments. We were anatomically identical to people today, yet our world would be unrecognizable to modern eyes. Now imagine that same span of time stretching forward, but instead of evolving on Earth, humanity lives among the stars. Isolated colonies scattered across solar systems. Generations born who never feel wind on their faces or rain on their skin. Children who grow up knowing Earth only as a distant blue dot in their sky. Scientists have confirmed that this duration is enough to fundamentally reshape our species through natural selection, genetic drift, and cultural divergence. What emerges after 10,000 years in space may no longer be recognizable as human. If you want to understand how the cosmos will sculpt our descendants into something entirely new, consider hitting that like button and subscribing. Now, settle in and get yourself comfortable. Let's begin. The moment humans leave Earth, they enter an environment that attacks the body from every direction simultaneously. This is not metaphor. This is not exaggeration for dramatic effect. Confirmed measurements from astronauts aboard the International Space Station reveal that space actively degrades nearly every biological system humans possess. Bones dissolve at rates that would terrify any physician. Muscles atrophy despite hours of daily exercise. Hearts shrink because they no longer need to work against gravity. Eyes deform under pressure shifts that evolution never anticipated. Immune systems malfunction in ways researchers are still struggling to understand. The human body evolved over hundreds of millions of years to function under very specific conditions, and space provides absolutely none of them. Understanding what 10,000 years would do to humanity requires first understanding what space does to individual humans over mere months and years. Only then can we begin extrapolating across the timescales where evolutionary forces start reshaping entire populations rather than just challenging individual survivors. Gravity shapes every single aspect of human physiology in ways most people never consider. Earth's gravitational pull measures approximately 9.8 meters per second squared at sea level. This constant downward force has been present for every moment of every day for every organism that has ever lived on this planet. Every bone in your skeleton, every muscle fiber in your body, every chamber of your heart, every fluid-filled space in your skull developed under this unceasing pull. Remove it, and the body begins systematically dismantling itself. Astronauts in microgravity lose approximately 1 to 2% of their bone mineral density per month. To put this in perspective, this rate exceeds the bone loss seen in severe osteoporosis patients by a factor of 10. An elderly person with dangerously brittle bones might lose 1% of bone density per year. Astronauts lose that much in 2 to 4 weeks. The weight-bearing bones in the legs and spine suffer the greatest losses because these structures evolved specifically to resist gravitational compression. They exist to hold you upright against the constant pull of Earth. Without that compression, the body interprets these dense calcium structures as metabolically wasteful and begins reabsorbing the minerals back into the bloodstream. Scientists have documented bone losses of 20% or more in astronauts who spent extended periods aboard orbital stations. But the skeletal system does not simply weaken uniformly like a crumbling building. It restructures itself according to the new mechanical environment it encounters. Bones become dramatically thinner in regions that no longer bear loads, while maintaining or even increasing density in areas that still experience stress from exercise equipment or locomotion. The skeleton literally reshapes itself to match its new functional demands. This remodeling follows precise biological logic. Bone tissue contains cells called osteoclasts that break down old bone, and osteoblasts that build new bone. Under normal Earth gravity, these processes balance each other, constantly refreshing the skeleton while maintaining its structural integrity. Mechanical stress signals tell the bones where to remain strong. In microgravity, those stress signals disappear from load-bearing regions. The osteoclasts keep working, but the osteoblasts receive no instructions to rebuild. The result is net bone loss concentrated precisely in the areas that would matter most upon return to gravity. Muscle atrophy follows a similar but even faster pattern. Postural muscles that normally work continuously against gravity begin wasting within days of reaching orbit. These are the muscles you never think about, the ones that keep you upright when standing, that stabilize your spine when sitting, that constantly fire in tiny corrections to maintain your position against the downward pull of Earth. In space, they have nothing to do. The body does not maintain unused tissue. The soleus muscle in the calf can lose up to 20% of its mass within just 2 weeks of microgravity exposure. This muscle specifically evolved to push blood back up toward the heart against gravity and to provide the spring in each step during walking. Without gravity, it becomes vestigial almost overnight. Even with rigorous exercise protocols requiring 2 hours of daily workouts using specialized resistance equipment, astronauts return to Earth significantly weaker than when they departed. The exercise slows the loss but cannot prevent it entirely. The cardiovascular system faces equally severe challenges that manifest in unexpected ways. On Earth, the heart must pump blood upward against gravity to reach the brain. This requires substantial muscular effort from the left ventricle and maintains cardiac fitness through constant work. Every heartbeat fights gravity. In microgravity, blood redistributes toward the head because gravity no longer pulls it toward the feet. Astronauts experience what they call puffy face bird leg syndrome as fluid shifts dramatically upward within hours of reaching orbit. The heart responds to this reduced workload by becoming smaller and weaker. Why maintain a powerful pump when the job has become easier? Studies of astronauts after long-duration missions reveal cardiac atrophy of up to 12%. The heart literally shrinks because it no longer needs to work as hard. This would be concerning enough on its own, but the changes extend throughout the entire circulatory system. Blood vessels in the legs lose their muscular tone because they no longer need to push blood back up toward the heart. The sophisticated network of valves and muscular contractions that prevent blood from pooling in the lower extremities becomes unnecessary. These systems begin degrading from disuse. Meanwhile, blood vessels in the upper body must cope with increased pressure. They never evolved to handle continuously. Perhaps most disturbing are the changes to vision that no one predicted before long-duration spaceflight became common. Approximately 70% of astronauts on missions lasting longer than 6 months experience measurable deterioration in their eyesight. The condition, now called spaceflight-associated neuroocular syndrome, involves flattening of the eyeball itself, swelling of the optic nerve where it enters the skull, and structural changes to the retina that can be observed directly through ophthalmologic examination. The precise mechanism remains under active investigation, but researchers suspect that increased intracranial pressure from fluid shifts plays a significant role. On Earth, gravity pulls cerebrospinal fluid downward during the day, creating pressure gradients that the skull and its contents evolve to accommodate. In microgravity, this fluid distributes differently, pressing against the back of the eyeballs with forces they were never designed to withstand continuously. Some vision changes persist permanently after return to Earth. Astronauts have reported lasting alterations to their eyesight following extended missions. This suggests that the eye, like many other organs, adapts structurally to the space environment in ways that cannot simply reverse upon returning to terrestrial conditions. The changes become built into the tissue itself. Radiation exposure presents an entirely different category of biological assault that operates independently of gravity but proves equally relentless. Earth's magnetic field and thick atmosphere shield surface life from the vast majority of cosmic radiation constantly bombarding our planet. This invisible shield deflects charged particles from the sun and blocks most of the high-energy cosmic rays that would otherwise sterilize Earth's surface. Space habitats lack this protection entirely. Astronauts aboard the International Space Station receive radiation doses approximately 50 to 100 times higher than people on Earth's surface, despite the station's relatively protected position within Earth's surface. Magnetic field. The station orbits low enough that Earth's magnetosphere provides partial shielding. Venture beyond this protective cocoon, and exposure increases dramatically. Measurements from spacecraft traveling to the moon recorded radiation levels hundreds of times higher than terrestrial background. The Apollo astronauts reported seeing flashes of light when they closed their eyes, caused by cosmic ray particles passing through their eyeballs and triggering their retinas directly. A journey to Mars would expose astronauts to radiation equivalent to receiving a full-body CT scan every 2 to 3 weeks for the entire duration of the trip, which could last over 2 years for a round-trip mission. This radiation damages DNA directly in ways the body cannot fully repair. Cosmic rays and solar particle events can break DNA strands, causing mutations that accumulate over time. Single-strand breaks can usually be fixed accurately by cellular repair machinery. Double-strand breaks are far more dangerous, often resulting in errors during repair that create permanent mutations or trigger cell death. The body possesses sophisticated mechanisms for detecting and repairing radiation damage, but these systems cannot fully compensate for chronic high-level exposure. Long-term consequences include dramatically increased cancer risk, as mutations accumulate in cells that divide frequently. Potential cognitive decline may result from damage to neurons and the brain's support cells. Perhaps most concerning for long-term space habitation, radiation may damage reproductive cells in ways that affect future generations. Mutations in eggs and sperm would be passed to children, potentially creating hereditary health problems unknown in Earth populations. The psychological stresses of space habitation compound these physical challenges in ways that researchers are only beginning to understand. Humans evolved as intensely social creatures living in varied natural environments filled with sensory richness. We need the sound of wind, the sight of horizons, the smell of earth and vegetation, the presence of diverse social contacts, the ability to walk away from conflicts. Space habitats provide none of these psychological necessities. Confined spaces smaller than most apartments house crews for months or years. Artificial lighting attempts to simulate day and night cycles but never quite matches the full spectrum and intensity variation of natural sunlight. Recycled air carries the same molecules through the same filters endlessly. Limited social contact with the same handful of people creates relationship dynamics that would strain any group. Extreme isolation from Earth, where help cannot arrive for months in an emergency, creates an existential vulnerability that gnaws at the subconscious. Studies of astronauts have revealed increased rates of anxiety, depression, and interpersonal conflict during long-duration missions. These are not weak-minded individuals. These are some of the most psychologically resilient humans ever selected through rigorous screening processes, yet the environment breaks them down regardless. The phenomenon of third quarter syndrome describes a predictable decline in mood and motivation approximately 3/4 of the way through a mission. Crews that performed excellently for months suddenly experience increased irritability, difficulty concentrating, and reduced enthusiasm for their work. The pattern appears so consistently that mission planners now anticipate and prepare for it. Sleep disturbances affect nearly all astronauts due to the absence of natural light-dark cycles and the constant background noise of life support systems. The human circadian rhythm evolved over millions of years to synchronize with the rising and setting of the sun. Artificial lighting signals fail to trigger the same hormonal cascades that regulate sleep on Earth. Many astronauts report sleeping poorly throughout their missions despite following carefully designed sleep schedules. These psychological stresses do more than affect mental well-being. They influence physical health in measurable ways. Chronic stress hormones like cortisol alter gene expression patterns, demonstrating that psychological states directly affect biological outcomes at the molecular level. Stress impairs immune function, accelerates cardiovascular deterioration, disrupts digestion, and interferes with the body's ability to repair damage from other sources like radiation. The environmental conditions of space habitats themselves present additional challenges beyond the absence of gravity and presence of radiation. Atmospheric composition must be carefully controlled, but the resulting air rarely matches Earth conditions exactly. Carbon dioxide levels aboard the International Space Station typically run 10 times higher than Earth's atmosphere. While not immediately dangerous at these concentrations, elevated carbon dioxide has been linked to headaches, cognitive impairment, and sleep disturbances that compound the other environmental stresses. Trace contaminants accumulate in closed-loop life support systems despite filtration efforts. Outgassing from materials slowly releases volatile organic compounds into habitat atmospheres. Human metabolic byproducts build up faster than scrubbers can remove them. Equipment off-gassing adds industrial chemicals to the air supply. Astronauts breathe air that has passed through mechanical filters and chemical scrubbers, lacking the biological and geological processing that characterizes terrestrial atmospheres. The smell of space stations reportedly takes weeks to stop noticing. Water in space habitats undergoes recycling processes that would horrify most Earth dwellers if they thought about them carefully. Urine, sweat, and atmospheric humidity are all reclaimed, processed through multiple filtration and purification steps, and returned to potable water supplies. The technology works remarkably well. Recycled water aboard the International Space Station meets safety standards more stringent than most municipal tap water, but it carries different mineral profiles and chemical signatures than naturally occurring water sources. These differences may have subtle but cumulative effects on human biology over multiple generations of continuous exposure. Microbial environments in space habitats differ dramatically from anything found on Earth. The diverse microbiome that colonizes human bodies evolved alongside terrestrial microorganisms over millions of years. Trillions of bacteria, fungi, viruses, and other microscopic organisms live in and on every human, performing essential functions for digestion, immune regulation, and even mood. Space habitats contain radically simplified microbial communities due to sterilization protocols and closed environments that prevent the normal exchange of microorganisms with the broader world. Astronaut microbiomes shift significantly during spaceflight, with documented changes in gut bacteria composition that may affect digestion, immune function, and even psychological well-being. Some of these changes reverse upon return to Earth. Others persist. The long-term consequences of raising generations of humans with fundamentally altered microbial companions remain entirely unknown. These individual stressors interact in complex ways that researchers are still working to understand. Radiation exposure may prove more damaging to immune systems already weakened by stress and microgravity. Bone loss may accelerate in the presence of altered gut bacteria that affect calcium metabolism. Psychological stress may impair the body's ability to repair radiation damage. Cardiovascular deconditioning may worsen sleep quality, which in turn may accelerate other forms of deterioration. The space environment creates a system of interconnected pressures that cannot be fully understood by examining each component in isolation. The stresses compound and multiply through feedback loops that make the whole worse than the sum of its parts. Now consider what happens when these pressures act not on individuals who return to Earth after months or years, but on populations living permanently in space across hundreds of generations. Evolutionary change requires three ingredients: variation, selection, and inheritance. Space provides all three in abundance. Genetic variation exists within any founding population because no two humans carry identical DNA. The space environment creates intense selective pressures, favoring certain traits over others. And reproduction passes both genetic and epigenetic information to subsequent generations, preserving the changes that proved beneficial. The key insight that makes space evolution plausible over 10,000 years is that evolutionary change does not require thousands of generations to produce measurable effects. Studies of isolated human populations right here on Earth have documented significant genetic changes occurring within just a few hundred years. The space environment would impose far stronger selective pressures than most terrestrial environments, potentially accelerating the pace of evolutionary change beyond anything observed in human history. Founder effects would begin reshaping space populations from the moment of colonization. Any group leaving Earth would carry only a subset of total human genetic diversity. Even a colony ship carrying 10,000 people would represent a genetic bottleneck compared to Earth's billions. This bottleneck would immediately make certain genetic variants more or less common than in the general human population. Rare alleles that happen to be carried by multiple founders might become dominant in the new population. Common alleles that no founder happened to carry would disappear entirely from that lineage forever. The size of founding populations matters enormously for subsequent evolution. Smaller populations experience more rapid genetic drift, where random chance causes allele frequencies to fluctuate dramatically between generations. Imagine a gene variant carried by 10% of a population of 10,000. Normal randomness in who reproduces successfully might shift that to 8% or 12% in the next generation. In a population of only 100, the same randomness might shift it to 2% or 20%. A colony of 1,000 individuals would experience much faster genetic change than one of 100,000, even if both faced identical environmental pressures. Isolation between colonies would accelerate divergence even further. If multiple human populations establish themselves in different space environments with limited migration between them, each population would evolve essentially independently. Different random mutations would arise and spread in each colony. Different selective pressures would favor different traits. Different founder effects would create different starting points. Within surprisingly few generations, these populations would begin accumulating genetic differences that distinguish them from each other and from the humanity that remained on Earth. This is not speculation about what might happen. It describes the documented process through which species diverge right here on Earth. Island populations, cave populations, isolated mountain valleys, and other geographically separated groups consistently evolve distinct characteristics within timeframes that seem remarkably short from an evolutionary perspective. Darwin's finches diverged into multiple species within roughly 1 million years. Cichlid fish in African lakes produced hundreds of species in similar timeframes. Isolated human populations have developed measurable genetic differences in mere thousands of years. 10,000 years represents approximately 400 human generations. This duration proved sufficient for dramatic changes in human populations that remained on Earth, connected by continuous gene flow and facing relatively mild selective pressures. Consider what happened to human populations that spread across Earth over the past 50,000 years. Groups that migrated to different continents developed distinct physical characteristics adapted to their local environments. Skin pigmentation varies dramatically with latitude, reflecting adaptation to different ultraviolet radiation levels. Populations near the equator maintain dark skin that protects against intense sunlight. Populations in northern latitudes evolved lighter skin that allows more vitamin D synthesis under weaker sun angles. Body proportions differ between populations from tropical and Arctic climates. Longer limbs dissipate heat more efficiently. Compact bodies conserve warmth better. Digestive capabilities vary based on ancestral diets in ways that persist today. Populations with long histories of dairy farming evolved lactose tolerance as adults, while populations without that history typically lose the ability to digest milk sugars after childhood. Populations with long histories of starch-heavy diets evolved additional copies of amylase genes to process those starches more efficiently. These changes occurred despite continuous gene flow between populations through migration, trade, and intermarriage. They occurred despite relatively weak selective pressures compared to what space would impose. A few % difference in reproductive success over many generations proved sufficient to reshape human biology in measurable ways. Space would impose selective pressures far stronger than different climates or diets. The individuals who survive and reproduce successfully in space environments would pass their genes to subsequent generations. Those who struggle with radiation damage, bone loss, cardiovascular deconditioning, vision deterioration, or psychological stress would be less likely to thrive and reproduce. They might survive. They might even have children, but on average they would have fewer children who themselves survive to reproduce. This selection would not require anyone to die dramatically before reproductive age. Evolution operates through differential reproduction, not just differential survival. Even small differences in fertility between individuals with different traits would compound across generations into enormous effects. If individuals carrying certain genetic variants have just 5% more children on average than those without, the favoured variants would become dramatically more common within a few dozen generations. Epigenetic changes would occur even faster than genetic selection because they do not require changes to DNA sequences. Epigenetics involves modifications to gene expression that can be inherited across generations. Methyl groups attached to DNA, modifications to histone proteins around which DNA wraps, and other molecular markers can turn genes on or off without altering the underlying genetic code. Environmental stresses trigger epigenetic changes that prepare offspring for similar conditions before those offspring are even conceived. Experiments have demonstrated that stress responses, metabolic adaptations, and even behavioral tendencies can be transmitted epigenetically from parents to children and grandchildren. Mice trained to fear a particular smell produce offspring and grand-offspring who show heightened fear responses to that smell, even though they never experienced the training themselves. The trauma of famine leaves epigenetic marks that affect metabolic regulation for multiple subsequent generations in humans. Spaceborn children would develop differently from Earthborn children even if they carried identical genetics. Their bones would form under different mechanical conditions from the very beginning of skeletal development. Their cardiovascular systems would calibrate to different fluid distributions during the critical periods when these systems organize themselves. Their eyes would develop in response to artificial lighting spectra and enclosed visual environments rather than natural sunlight and open horizons. These developmental differences would not immediately become hereditary in the strict genetic sense, but they would establish the phenotypic baseline from which selection operates. Natural selection does not see genes directly. It sees bodies, behaviors, and reproductive outcomes. The environmental conditions that shape development would influence which genetic variants proved advantageous, creating indirect pathways through which environment shapes evolutionary trajectory. The interplay between genetic change, epigenetic inheritance, and developmental plasticity would create complex patterns of adaptation across generations. Some changes would appear quickly as bodies responded to immediate environmental conditions through existing plasticity mechanisms. Others would accumulate slowly as natural selection gradually shifted allele frequencies in the underlying genetic code. Still others would emerge through interactions between genetic predispositions and environmental triggers that neither factor alone would produce. What begins as individual physiological responses to space conditions would gradually become population-level characteristics encoded in genes and passed automatically to offspring. The boundary between environmental effect and genetic adaptation would blur across generations as traits that initially required specific environmental triggers became genetically fixed in populations where those triggers were universal. This transformation would not occur uniformly across all space-dwelling populations. Different space environments would impose dramatically different selective pressures that would push human biology in different directions. Colonies on rotating habitats simulating Earth normal gravity would face different challenges than stations maintaining microgravity throughout. Settlements orbiting close to stars would contend with different radiation profiles than those traveling through interstellar space. Underground colonies on airless moons would experience different atmospheric conditions and social configurations than orbital platforms housing thousands. Each distinct environment would create its own selective regime. Each would favor different traits. Each would accumulate different mutations and fix different adaptations. The diversity of space environments available for human habitation would generate corresponding diversity in the human forms that emerged to inhabit them. What begins as a single species spreading into space would gradually fragment into populations adapted to their specific cosmic niches, like Darwin's finches radiating across the Galapagos, like cichlids diversifying through the African lakes, like mammals filling ecological roles after the dinosaurs vanished. Humanity would diversify to fill the varied environments the cosmos offered. 10,000 years from now, The descendants of space colonists might look back on the earliest astronauts the way modern humans look back on our ancient ancestors. They would see bodies and minds adapted to an environment their own biology could barely tolerate. They would recognize the kinship while marveling at how much had changed. They would understand that the relentless pressures of space did not merely challenge humanity, but fundamentally reshaped what humanity means. The transformation begins the moment the first permanent space dweller is born beyond Earth. Every subsequent generation carries the process forward, each small change building on those that came before. The environment selects, genetics responds, culture adapts, and slowly, imperceptibly at first, then unmistakably, a new kind of human begins emerging from the cosmic crucible. The pressures described here are just the beginning. They establish why change would occur and how quickly it might proceed. But what exactly would those changes look like as they accumulated across centuries and millennia? What would space do to human bones, hearts, eyes, and immune systems over timescales long enough for evolution to work its slow transformations? The biology of becoming awaits. The human skeleton tells a story written in calcium and collagen. Every bone records the mechanical forces it has experienced throughout life, strengthening where stressed and weakening where neglected. This continuous remodeling allows bones to adapt to changing demands within a single lifetime. A tennis player develops denser bone in their dominant arm. A bedridden patient loses skeletal mass within weeks. The skeleton is not a static scaffold, but a living tissue constantly rebuilding itself according to the signals it receives. Across generations in space, this same biological plasticity would enable transformations far more dramatic than anything a single lifetime could produce. What begins as individual adaptation would become hereditary modification, as natural selection favored the genetic variants underlying the most successful responses. Confirmed research on bed rest studies, paralysis patients, and astronauts demonstrates conclusively that bones require mechanical loading to maintain their density and structure. Remove the loading, and bones begin dissolving within days. The cellular machinery responsible for this dissolution operates continuously throughout life. Under normal circumstances, bone-building cells called osteoblasts balance the bone-destroying activity of osteoclasts. Mechanical stress signals tell the osteoblasts where to work. Without those signals, the balance tips toward destruction. The body does not understand why the mechanical load disappeared. It cannot distinguish between floating in space and lying motionless in a hospital bed. It simply responds to the absence of stress signals by reducing investment in structural material that appears to serve no function. From the body's perspective, maintaining dense leg bones in microgravity wastes precious calcium and metabolic energy on useless architecture. In the first generations of permanent space habitation, this aggressive bone remodeling would create serious health challenges requiring constant technological intervention. Individuals would need extensive exercise protocols using resistance equipment that simulates gravitational loading. Pharmaceutical interventions might slow calcium loss through drugs that inhibit osteoclast activity. Some colonies might provide periodic artificial gravity exposure through rotating sections or centrifuge facilities. Many individuals would struggle despite these efforts. Bones would fracture more easily. Spinal problems would proliferate. The skeletal system simply was not designed for the environment these pioneers would inhabit. Natural selection would begin operating immediately on this struggling population. Individuals whose bones retain density more efficiently under reduced gravity conditions would experience fewer fractures, less chronic pain, and better overall health. These advantages would translate into reproductive success through multiple pathways. Healthier individuals would be more attractive as mates. They would have more energy to devote to child rearing. They would live longer and potentially reproduce more times. Their children would be more likely to survive to adulthood if they inherited the favorable skeletal traits. Small genetic variations affecting calcium metabolism would suddenly matter enormously. Variants in genes controlling how efficiently the intestines absorb dietary calcium, how effectively the kidneys retain calcium rather than excreting it, how sensitively bone cells respond to mechanical signals. how quickly bone matrix mineralizes after formation. These subtle genetic differences that might never matter on Earth would become crucial determinants of success in space. Structural protein production would face similar selection. Collagen forms the organic matrix upon which bone minerals deposit. Different collagen variants produce matrices with different properties. Some might prove more resistant to degradation in the absence of mechanical loading. Others might maintain structural integrity with lower mineral content. The genes encoding these proteins would shift in frequency as variants conferring advantages spread through space populations. Over hundreds of generations, these small advantages would compound into substantial population-level changes. The genes promoting efficient bone maintenance in microgravity would spread through space populations, while those associated with rapid bone loss would decline in frequency. After several thousand years, the average space human would possess bones better adapted to their environment than any Earth human could achieve through exercise or medication alone. The adaptation would be built into their biology from conception. But adaptation does not simply mean maintaining Earth-normal bones under abnormal conditions. Evolution rarely works by preserving ancestral forms against environmental pressure. Evolution finds creative solutions that often look nothing like what came before. Rather than fighting endlessly against the pressures of microgravity, space-adapted skeletons might embrace fundamentally different architectures that work with the new environment rather than against it. Studies of aquatic mammals reveal how dramatically evolution can restructure skeletal systems when mechanical environments change. Whale ancestors were four-legged land mammals with dense bones suited for supporting body weight against gravity. Over millions of years of adaptation to marine life, those bones transformed completely. Modern whale bones are spongy and porous, filled with oil rather than marrow, providing buoyancy rather than structural support. The mechanical requirements of swimming through water differ so completely from walking on land that evolution rebuilt the entire skeleton according to new principles. A similar transition might occur in space-dwelling humans, whose bodies no longer require the robust skeletal framework that terrestrial life demands. Earth-human legs contain the thickest, densest bones in the body because they must support our entire weight with every step, while also absorbing the shock of impact with the ground. The femur, the thigh bone, can withstand forces of several thousand pounds before fracturing. This extraordinary strength requires extraordinary investment in calcium, collagen, and metabolic maintenance. In microgravity, legs serve entirely different functions. They become additional limbs for grasping, pushing off from surfaces, and maneuvering through three-dimensional space. They no longer bear weight in the terrestrial sense. The selective pressure maintaining their massive architecture would disappear. Instead, selection might favour lighter, more flexible leg bones that consume fewer resources while providing adequate support for microgravity movement patterns. Over thousands of years, space-human leg bones might thin considerably from their Earth proportions. The massive femoral shaft might become more slender, maintaining enough strength for propulsion and manipulation without the excess capacity needed for weight-bearing. The tibia and fibula might fuse or simplify, as occurred in many bird lineages when flight reduced requirements for terrestrial locomotion. Foot bones might elongate and become more prehensile, better suited for grasping holds than for supporting standing weight. Arms and hands would face opposite selective pressures. In microgravity, the upper limbs become primary means of propulsion and manipulation. Astronauts on the International Space Station move primarily by pulling themselves along handholds, pushing off from surfaces with their arms, and manipulating equipment with their hands. These activities place demands on the upper limb skeleton that differ substantially from Earth patterns. Finger bones might strengthen and elongate over generations as hands became more important for locomotion. The arms might develop greater range of motion at the shoulder and elbow to facilitate three-dimensional maneuvering. The clavicles might restructure to support different patterns of force transmission. These changes would accumulate gradually, each generation slightly better suited to microgravity movement than the last. The spine presents particular evolutionary interest because it must function so differently in space than on Earth. Terrestrial human spines evolved to support upright posture against gravity while protecting the spinal cord and allowing flexible movement. The characteristic S-curve of the human spine distributes gravitational loads efficiently while positioning the head directly over the pelvis. The intervertebral discs act as shock absorbers between vertebrae, compressing under load during the day and expanding during sleep when the body lies horizontal. In microgravity, this entire mechanical system loses its functional rationale. Astronauts actually grow taller in space as their intervertebral discs expand without gravitational compression. This expansion can cause back pain as spinal ligaments stretch beyond their normal range. The muscles that maintain spinal posture on Earth have nothing to do in space, and they atrophy rapidly. Space-adapted spines might evolve toward greater flexibility at the cost of load-bearing capacity. The vertebrae might become smaller and more uniform, without the progressive size increase from cervical to lumbar regions that reflects gravitational loading on Earth. The intervertebral discs might transform into different structures better suited for a life without compression. The spine might curve differently or not at all, adopting whatever configuration proves most functional for three-dimensional movement. The pelvis would undergo equally dramatic restructuring as its mechanical function changed completely. The human pelvis represents a remarkable evolutionary compromise between the requirements of bipedal locomotion and the requirements of giving birth to large-brained infants. The pelvic bowl transmits forces between the legs and spine during walking, while also providing the birth canal through which babies must pass. In microgravity, bipedal locomotion becomes irrelevant. The force transmission function of the pelvis disappears. Selection pressure would shift entirely toward reproductive function and whatever new mechanical demands microgravity movement imposed. The pelvis might widen or narrow, flatten or deepen, according to which configurations produced the most successful births and the most effective movement in the new environment. After 10,000 years, space human pelvic anatomy might bear little resemblance to terrestrial forms. These skeletal changes would occur so gradually that no one would notice them happening. Parents would not observe that their children's bones differed from their own. Each generation would seem perfectly normal within its own context. Only comparison across centuries would reveal the transformation. Only measurements against historical records would demonstrate how far the population had drifted from its founding stock. Paleontologists of the far future studying space human remains would recognize them as descendants of terrestrial humans. The fundamental body plan would remain recognizable. The same bones would be present, arranged in the same general pattern, but the proportions, densities, and detailed structures would differ enough to mark these beings as distinct from any population that had ever lived on Earth. They would represent a new chapter in human skeletal evolution, written by the selective pressures of cosmic environments. Cardiovascular adaptation would follow parallel but distinct evolutionary trajectories. The heart and blood vessels evolved over hundreds of millions of years to overcome gravity's constant downward pull on blood. Every aspect of the circulatory system reflects this fundamental challenge. The muscular walls of arteries resist blood pressure created by the pumping heart. The one-way valves in veins prevent blood from falling backward between heartbeats. The baroreceptors in the neck detect pressure changes when posture shifts and trigger reflexes that maintain cerebral blood flow. Without gravitational challenge, this entire system would reorganize itself according to different functional requirements. The elaborate mechanisms for maintaining blood pressure while standing would become unnecessary overhead. The powerful left ventricle designed to pump blood upward to the brain would find its job dramatically simplified. The complex venous return system in the legs would lose its functional purpose entirely. Early space generations would experience the cardiovascular deconditioning already documented in astronauts returning from long missions. Their hearts would shrink from disuse, their blood vessels would lose muscular tone, their baroreceptor reflexes would weaken. If these individuals returned to Earth, they would experience orthostatic intolerance, becoming dizzy or fainting when standing up as blood pooled in their legs faster than their deconditioned systems could compensate. For permanent space inhabitants, however, This deconditioning would not represent dysfunction but rather appropriate calibration to the actual environment. A heart powerful enough to pump blood against Earth gravity wastes energy in an environment where gravity poses no obstacle. Blood vessels toned for terrestrial circulation maintain unnecessary muscular mass. The deconditioned state that poses problems for returning astronauts would become the baseline normal for populations that never return. Over generations, selection would favor cardiovascular configurations optimized for microgravity circulation, rather than those designed to fight against absent gravity. Blood pressure regulation systems might simplify dramatically. The complex feedback loops involving baroreceptors, the autonomic nervous system, and vascular smooth muscle evolved to handle rapid postural changes in gravity. In an environment without such changes, simpler regulation might suffice. The heart itself might adopt different chamber proportions. The left ventricle, which performs the heavy lifting of systemic circulation, is much more muscular than the right ventricle in Earth humans. This asymmetry reflects the higher pressures required to push blood throughout the body compared to the lower pressures of pulmonary circulation. In microgravity, with gravitational resistance removed, the pressure differential between systemic and pulmonary circulation might change. Heart evolution might follow. Fluid distribution patterns would stabilize in new configurations unlike anything seen in terrestrial humans. On Earth, blood and interstitial fluid pool in the lower body during upright hours, then redistribute more evenly during sleep. This daily cycling shaped the evolution of human fluid regulation systems. In microgravity, no such cycling occurs. Fluids distribute according to different principles, accumulating in the upper body in ways that Earth humans find uncomfortable, but space-adapted humans might consider normal. The face puffiness and leg thinning that afflicts Earth astronauts represents their body's failed attempt to reach a new equilibrium while still carrying terrestrial programming. True space humans, after generations of selection, would achieve genuine equilibrium. Their bodies would expect and accommodate the fluid distributions that space environments produce. Their faces would not look puffy to themselves because their developmental programs would build faces anticipating those fluid levels. Their legs would not look thin because leg development would calibrate to expected conditions. Vision changes present particular evolutionary interest because eyes directly affect survival and reproduction in obvious ways. Clear vision enables effective work, safe movement, and accurate assessment of potential mates and threats. The vision deterioration that afflicts a majority of long-duration astronauts would impose strong selective pressure against whatever genetic and developmental factors contribute to spaceflight-associated neuroocular syndrome. The syndrome appears to involve increased intracranial pressure from fluid redistribution, which pushes against the back of the eyeballs and distorts their shape. Individuals whose skull anatomy, cerebrospinal fluid dynamics, or eyeball structure better resisted this pressure would maintain clearer vision. Their superior visual function would translate into reproductive advantages through multiple pathways. They would be more effective workers and leaders. They would make fewer dangerous mistakes. They would appear healthier and more capable to potential mates. Over generations, the population would shift toward greater resistance to pressure-induced vision changes. Skulls might develop different configurations that better accommodated altered fluid distributions. Eyeball structure might change to resist deformation. The entire complex of anatomical features contributing to the syndrome would face selective pressure toward configurations that avoided the problem. Beyond simply maintaining Earth normal vision, space environments might select for entirely different visual capabilities. Artificial lighting in space habitats differs fundamentally from natural sunlight in spectrum, intensity, and temporal patterns. The sun produces light across a broad spectrum peaking in the green-yellow range that human vision evolved to exploit. Artificial lights produce narrower spectra, determined by their generating technology. Eyes optimized for sunlight might not represent optimal solutions for artificial illumination. Color perception might shift over generations to match the spectral output of common artificial light sources. The three types of cone cells in human retinas, sensitive to different wavelength ranges, could evolve different peak sensitivities that better discriminated objects under artificial lighting conditions. The result would be color vision adapted to the actual light environment, rather than the ancestral solar spectrum. Night vision might become less important in habitats with continuous artificial lighting. The rod cells that enable vision in dim light require metabolic resources to maintain. In environments never experiencing natural darkness, these resources might be redirected toward other visual capabilities. Space humans might eventually lose significant night vision capacity while gaining enhanced function under artificial light conditions. Distance vision might become less acute in enclosed environments where nothing exists beyond a few hundred meters. The ability to resolve fine details at great distances evolved for hunting, navigation, and predator detection across open landscapes. Space habitats contain no distant horizons. Selection might favor near vision capabilities at the expense of distance acuity that serves no function. The immune system faces perhaps the most complex adaptive challenges because immunity involves such intricate interactions between genetics, development, environment, and experience. Human immune systems evolved in constant contact with diverse microbial communities that changed with seasons, locations, and life stages. Space habitats provide none of these evolutionary familiar conditions. The documented changes in astronaut immune function reveal systems struggling to calibrate themselves without expected inputs. Some responses become overactive, producing inflammation and autoimmune tendencies in the absence of genuine threats. Other responses become suppressed, reducing defense against pathogens that might still pose danger. The immune system expects certain challenges and prepares for them whether or not they materialize. Without the expected microbial environment, this preparation misfires. Space habitats maintain relatively sterile conditions compared to terrestrial environments. Extensive filtration, sterilization protocols, and limited biological diversity reduce microbial populations to fractions of Earth normal levels. The immune systems of inhabitants lack the training that exposure to diverse microorganisms normally provides. They remain naive to pathogen types they might encounter, and overreact to harmless stimuli they misidentify as threats. Over generations, immune systems would adapt to these new conditions through selection, favoring appropriate calibration. Individuals whose immunity functioned well in low pathogen environments, neither overreacting nor underreacting to the actual microbial challenges present, would enjoy health advantages translating into reproductive success. The population would gradually shift toward immune configurations suited to space habitat conditions. This adaptation carries significant risks that might only become apparent upon contact with other populations. Space-evolved immune systems might lose defenses against pathogens that no longer threaten isolated space communities. If contact with Earth or other colonies expose space humans to unfamiliar microorganisms, their streamlined immune systems might struggle to mount effective responses. Diseases that Earth populations handle routinely might prove devastating to immune systems that had never encountered anything similar. This vulnerability would create powerful selective pressures during any reconnection event. Space populations making contact with Earth or other colonies might experience epidemic mortality as unfamiliar pathogens swept through immunologically naive populations. This pattern repeated throughout Earth history when isolated populations reconnected after long separation. The consequences for space humans might prove even more severe, given the more extreme isolation and more complete immune system simplification that space habitation might produce. Reproductive biology would undergo its own suite of transformations as human reproduction adapted to conditions no Earth organism ever experienced. Human reproduction evolved for terrestrial life over hundreds of millions of years. Every aspect of conception, gestation, and birth assumes gravitational conditions. Sperm swim toward eggs using flagella that evolved to work in terrestrial fluids under terrestrial gravity. Embryos develop through cell division patterns that assume gravitational orientation. Fetuses position themselves for birth using gravity as a reference. The birth process itself involves the descent of the baby through the birth canal under gravitational assistance. No human has ever been conceived, gestated, or born in space. animal studies suggest that reproduction in microgravity faces significant challenges. Sperm motility patterns change in ways that might affect fertilization success. Embryonic development can be disrupted at various stages. The absence of gravitational cues might impair normal developmental processes that rely on orientation. The first generations conceived and born in space would essentially conduct uncontrolled experiments in extraterrestrial human development. Some individuals would develop normally despite the unusual conditions. Others would experience developmental problems ranging from minor abnormalities to severe defects incompatible with life. The genetic backgrounds producing more viable offspring under space conditions would become more common in subsequent generations, as their bearers contributed more children to the population. Developmental patterns might gradually shift to accommodate space realities. The timing of embryonic stages might change. The signals triggering various developmental processes might evolve alternatives to gravity-dependent cues. The entire choreography of human development, refined over millions of years of terrestrial evolution, might reorganize itself around different environmental inputs. Birth mechanics would face particular selective pressure, since birth represents such a critical choke point in reproductive success. Difficult births that would be managed through medical intervention on Earth might prove fatal in space colonies with limited medical resources. Selection would strongly favor whatever anatomical configurations produced easier births under space conditions. Pelvic shapes, fetal sizes, and the flexibility of birth-related tissues might all evolve toward configurations that reduced birth complications. Sexual selection adds another evolutionary dimension that would reshape space populations in ways having nothing to do with survival per se. Human mate choice involves complex preferences shaped by both biological instincts and cultural learning. These preferences influence who reproduces with whom, and thus which genes pass to subsequent generations. In the novel social environments of space colonies, different traits might become attractive. Physical characteristics associated with space competence might gain appeal. Individuals who moved gracefully in microgravity, who maintained health despite environmental challenges, who demonstrated valuable technical skills, might become preferred mates regardless of whether those traits would have conferred any advantage on Earth. Cultural values prioritizing colony survival might favor personality types suited to confined group living. The aesthetic preferences that influence reproduction would themselves evolve in response to space conditions. Metabolism and nutrition would face selective pressures related to the unique constraints of space food production. Growing food in space requires substantial resources. Every calorie consumed must be produced through agricultural systems that demand energy, water, nutrients, and space. Recycling efficiency becomes paramount when nothing can be imported from outside the closed system. Waste becomes intolerable when it represents lost resources that cannot be replaced. Individuals whose bodies extracted nutrients more efficiently from limited food supplies would enjoy reproductive advantages during resource-constrained periods that every space colony would inevitably experience. Bodies that maintained function on fewer calories, that wasted less of what they consumed, that tolerated nutritional imbalances better, would prove more successful than metabolically expensive bodies requiring abundant, diverse nutrients. Over time, space human metabolism might become significantly more efficient than terrestrial metabolism. Digestive systems might adapt to extract maximum nutrition from whatever food sources proved most practical for space agriculture. Perhaps algae or insects or hydroponic vegetables would dominate space diets, and digestive systems would optimize for these specific foods rather than maintaining the generalist capabilities that served human ancestors eating varied wild diets. Nutritional requirements themselves might shift based on available nutrient profiles. If certain vitamins or minerals proved difficult to produce in space agricultural systems, selection might favor individuals requiring less of those nutrients. If others proved abundant, tolerance for higher levels might evolve. The metabolic changes would create bodies suited to space sustenance, but potentially unable to thrive on Earth normal diets that space-adapted systems could not properly process. Genetic drift would compound these selective pressures in reshaping space populations. In small, isolated communities, random chance significantly influences which genes pass to subsequent generations regardless of their survival value. A brilliant individual might die in an accident before reproducing, removing their genes from the population. A mediocre individual might happen to have many children who all survive, spreading their genes widely. Over generations, these random events accumulate into population-level genetic changes. Computer simulations of genetic drift in population sizes typical of early space colonies predict rapid changes in allele frequencies even without selective pressure. A colony of a few thousand individuals would experience significant genetic drift within just a few hundred years. Combined with the intense selection imposed by space conditions, drift would accelerate the divergence of space humans from their Earth origins in unpredictable ways. Different colonies would drift in different directions based on their unique founding populations and the specific random events each experienced. A beneficial mutation arising in one colony might spread throughout that population while never appearing in others simply because mutations occur randomly. Genetic bottlenecks during colony founding events, when only a small group established a new settlement, or during disaster events that killed large portions of a population, would further shape each colony's distinctive genetic profile. The isolation between colonies would prevent the gene flow that normally slows divergence between populations. Unlike Earth populations, which exchange genes through migration and intermarriage across continents throughout history, space colonies separated by travel times measured in years or decades would evolve essentially independently. Each colony would become its own evolutionary experiment, drifting and adapting according to its specific circumstances. After 10,000 years of isolation, the genetic differences between separated space populations might approach or exceed those between the most divergent human populations on Earth today. Certain colonies might accumulate changes affecting not just superficial features, but fundamental developmental processes. The boundaries of what counts as the same species could begin blurring at the edges. This biological transformation would occur without anyone planning or intending it. No one would set out to create new human variants. The changes would simply accumulate, generation after generation, as the relentless mathematics of evolution did what it always does when populations face new environmental challenges and become isolated from each other. The cosmos would sculpt humanity according to its own impersonal requirements, indifferent to any human preferences about the outcome. Each individual change would seem small and unremarkable to those experiencing it, a slight increase in bone density efficiency that no one could perceive directly. Marginally better radiation repair mechanisms invisible to everyone, except specialized medical equipment. Subtly altered cardiovascular regulation that felt completely normal to those possessing it. Trivial differences that no one would notice between parents and children, because the differences would fall within the normal range of human variation. But these trivial differences would compound across centuries and millennia, building toward transformations that only historical perspective could reveal. Like interest compounding in a savings account, small percentage changes applied generation after generation would accumulate into substantial totals. The space humans of the far future would differ from their founding ancestors by amounts that would seem impossible when viewing any single generational step. The descendants of space colonists would not think of themselves as evolved or transformed. They would simply be human, as they understood the term. Their bodies would feel normal to them because those bodies would be all they had ever known. Their hearts would beat at rates that felt right. Their bones would support activities that felt natural. Their eyes would see in ways that matched their expectations about how seeing should work. Only comparison with historical records or contact with Earth populations would reveal how much had changed. Only measurement against baseline data from the founding generation would demonstrate the drift. Only meeting their terrestrial cousins would make apparent how differently the two populations had developed during their long separation. And yet change would be undeniable once comparison became possible. The biology of space humans would differ from Earth humans in ways both subtle and profound. They would carry different frequencies of genetic variance, weighted by selection towards space-favorable traits. They would develop along different trajectories, calibrated by epigenetic programming to space conditions. They would possess capabilities and vulnerabilities unknown to their terrestrial ancestors. 10,000 years of cosmic selection would write its story in their flesh, their bones, their blood, their cells, their very DNA. They would be the biological record of humanity's adaptation to the universe beyond Earth, and their minds would prove equally transformed by the cosmic environment in ways the next chapter must explore. The human brain evolved to navigate African savannas, not infinite voids. Every cognitive system we possess reflects ancestral challenges that have nothing to do with space travel. Our spatial reasoning assumes solid ground beneath our feet and a reliable down direction that never changes. Our social instincts expect tribes of perhaps 150 individuals whose faces we recognize and whose reputations we track across lifetimes. Our emotional responses calibrate to threats and opportunities that existed millions of years before the first rocket launched. Fear of heights made sense when falling meant death. Claustrophobia motivated escape from enclosed spaces where predators might trap us. Loneliness drove us back toward groups where survival odds improved. Placing this ancient neural architecture in radically alien environments creates immediate psychological strain that no amount of training fully eliminates. The astronauts who have ventured beyond Earth report consistent patterns of cognitive and emotional adjustment, not all of them comfortable and not all of them reversible. Over generations, these individual responses would become population-level characteristics, as space environments selected for minds suited to their demands, rather than minds carrying the full burden of terrestrial programming. Spatial cognition would transform so fundamentally that Earth humans and space humans might eventually perceive physical reality in incompatible ways. Human perception of up and down derives primarily from the inner ear's vestibular system, which uses gravity to determine orientation. Tiny calcium carbonate crystals called otoliths rest on beds of sensory hair cells. Gravity pulls these crystals downward, bending the hair cells and sending signals to the brain indicating which direction is down. This system operates continuously and unconsciously, providing the foundation upon which all spatial reasoning builds. In microgravity, this system provides no useful information whatsoever. The otoliths float rather than pressing against their sensory beds. The hair cells send confused or absent signals. The brain receives no gravitational orientation data from the system that evolved specifically to provide it. This creates not merely confusion, but a fundamental disconnect between neural expectations and environmental reality. The brain must learn to rely entirely on visual cues for spatial orientation, a challenging adaptation that causes significant disorientation during the transition period. Astronauts report weeks of adjustment during which the visual world seems unstable. Nausea is common, and simple movements require conscious attention that would be automatic on Earth. The brain essentially rewires itself to process spatial information through different channels than those evolution prepared. Astronauts who spend extended periods in space report that they gradually develop new spatial frameworks for navigating three-dimensional environments without gravitational reference. Some describe arbitrary assignments of up and down within habitats based on lighting, direction, furniture orientation, or simple convention. The direction your head points when you enter a module becomes up. The direction of the main lighting fixtures becomes up. These assignments feel natural after sufficient exposure, even though they bear no relationship to any physical force. Others develop what they describe as true, three-dimensional navigation abilities that feel natural but differ fundamentally from earthbound spatial reasoning. They stop thinking in terms of up and down entirely, instead conceptualizing space as a volume through which they move without preferential orientation. Walls become floors, become ceilings, depending on how you approach them. Every surface offers potential footing. The very concept of falling loses, meaning when there is nowhere to fall to. These individual adaptations hint at what population-level changes might look like across generations. Children born and raised in microgravity would never develop the gravity-based spatial frameworks that all Earth humans share, regardless of culture or experience. Their brains would wire themselves from the very beginning of development for three-dimensional movement without preferential orientation. The vestibular system would still exist, still develop, but would calibrate to conditions where its primary evolved function serves no purpose. Research on neural plasticity demonstrates that the brain allocates resources according to functional demands. Regions that process frequently used information expand at the expense of less used areas. Musicians develop larger auditory cortices. Taxi drivers develop larger hippocampi for spatial memory. Individuals who lose one sense often develop enhanced capabilities in remaining senses as neural territory gets reallocated. Space human brains might restructure significantly to support three-dimensional spatial reasoning more efficiently than terrestrial brains can achieve. Brain regions devoted to vestibular processing might shrink or repurpose themselves for other functions when vestibular input carries no useful information. Areas supporting visual spatial integration might expand to handle the increased demands of navigating without gravitational reference. The neural architecture underlying navigation and spatial memory might reorganize according to principles that evolution never had opportunity to explore on Earth. The hippocampus, a brain structure crucial for spatial memory and navigation, contains specialized cells called place cells and grid cells that create cognitive maps of environments. On Earth, these maps are essentially two-dimensional, with height as a secondary consideration. We think of locations as positions on a surface, with elevation added as needed. Space-adapted hippocampi might develop truly three-dimensional mapping capabilities, creating spatial memories organized according to principles unknown in any terrestrial brain. These neurological changes would confer significant advantages for microgravity navigation, while potentially creating profound disorientation if space-adapted individuals ever experienced gravity. A brain wired from infancy for three-dimensional orientation without preferential direction might struggle to adapt to an environment where down exists as a constant, inescapable reality. The first space humans to visit Earth after generations of orbital living might find gravity not merely uncomfortable but cognitively overwhelming, their spatial processing systems unable to handle the asymmetry that terrestrial brains take completely for granted. Time perception would shift in environments lacking the natural temporal cues that shaped human consciousness over evolutionary time. Earth humans evolved with circadian rhythms entrained to the 24-hour day-night cycle created by our planet's rotation. These rhythms are not merely habits we could break with sufficient willpower. They are written into the molecular machinery of every cell, governing gene expression, hormone production, metabolic activity, and countless other biological processes according to daily patterns established over hundreds of millions of years of evolution under a cycling sun. Sunlight triggers these biological processes through specialized receptors in the eyes that detect blue wavelengths characteristic of daylight. These receptors connect to the suprachiasmatic nucleus, a tiny brain region that serves as the body's master clock, coordinating the circadian timing of tissues throughout the body. Morning light resets this clock daily, synchronizing internal rhythms with external conditions. The entire system assumes that light means day, darkness means night, and the cycle repeats reliably every 24 hours. Space habitats provide no natural day-night cycle. Orbital stations experience 16 sunrises and sunsets every 24 hours as they circle Earth. Deep space missions receive constant illumination that varies with distance from stars but not with any regular cycle. Artificial lighting can simulate solar patterns, but the simulation never perfectly matches natural conditions in spectrum, intensity, timing, or the subtle variations that terrestrial life uses to track time's passage. The absence of seasonal variation removes additional temporal landmarks that terrestrial humans use to organize experience. Spring, summer, autumn, winter create a rhythm of change that marks years and provides psychological anchoring. Weather changes from day to day create variation that structures time perception. Even the movement of sun and stars across the sky provides cues that space habitats cannot replicate. Time in space tends toward undifferentiated sameness that humans find psychologically challenging. Studies of astronauts reveal significant disruptions to circadian rhythms during spaceflight, even when artificial lighting attempts to maintain Earth-like cycles. Sleep quality declines despite adequate time allocated for rest. Cognitive performance fluctuates unpredictably, rather than following the predictable daily patterns typical on Earth. The subjective experience of time's passage changes in ways astronauts struggle to articulate. Days blur together. Weeks seem simultaneously too long and too short. The psychological anchoring that comes from natural temporal rhythms loosens. These disruptions contribute substantially to the psychological challenges of long-duration missions. Humans need temporal structure to maintain psychological equilibrium. We need to feel that time moves, that days differ from each other, that we progress through meaningful intervals toward anticipated events. Without these feelings, motivation suffers, mood deteriorates, and cognitive function declines even when nothing else has changed. Space-adapted populations might develop different circadian biology, better suited to artificial environments than the solar-calibrated systems all terrestrial humans share. Sleep patterns might fragment into multiple shorter periods rather than one consolidated nighttime block. This polyphasic sleep, rare but not unknown on Earth, might prove more functional in environments where work schedules and life support demands do not respect arbitrary day-night divisions. Hormonal cycles that currently synchronize with light exposure might decouple from external cues entirely and operate according to internal rhythms that need no environmental resetting. The molecular clocks governing cellular function might adjust their periodicity to match whatever schedule proves most functional for space living, whether that matches Earth's 24 hours or some entirely different interval. The subjective experience of time might calibrate to institutional schedules and task completion rather than astronomical cycles. Space humans might not feel that days pass so much as that work shifts begin and end, that sleep periods arrive and conclude, that meals punctuate ongoing activities. Their time perception might center on social rhythms rather than cosmic ones, creating a fundamentally different relationship with temporality than any Earth human has ever experienced. Social cognition faces perhaps the greatest adaptive challenges, because human social instincts are so specific to conditions that space environments cannot provide. Human social brains evolved for life in small bands, where everyone knew everyone else by face, voice, history, and reputation. These bands typically numbered around 150 individuals, a figure called Dunbar's number after the anthropologist who identified it. Our brains can maintain meaningful social relationships with roughly this many people before the cognitive load becomes unmanageable. Within these ancestral bands, reputation management was essential for survival. Individuals who cheated, who failed to reciprocate, who took more than they gave, faced social consequences that could mean death in environments where group membership determined survival. The human brain evolved exquisite sensitivity to social dynamics, to fairness and unfairness, to alliance and betrayal, to the subtle signals of trustworthiness and threat that governed ancestral social life. The social structures of space colonies might differ dramatically from these ancestral patterns in ways that would reshape social cognition over generations. Small colonies would create intense social environments where the same individuals interact constantly across all life domains without possibility of escape. Your work colleagues would also be your neighbours, your recreation partners, your potential mates, your children's teachers, and possibly your relatives. The social compartmentalization that characterizes modern urban Earth life, where you can maintain separate social circles that never intersect, would be impossible in communities of a few hundred or a few thousand individuals. This social intensity might favor different personality profiles than those succeeding in diverse Earth environments where one can always find new social niches if current ones become uncomfortable. Individuals highly skilled at conflict avoidance and social harmony maintenance might thrive in colonies where disagreements cannot be escaped through physical separation. Those requiring greater social variety, more privacy, or more opportunity for fresh starts might struggle psychologically in environments offering none of these. Over generations, space populations might shift toward more communal personality types suited to their inescapable social proximity. The trait distributions that characterize Earth humanity, with its broad range from extreme introversion to extreme extroversion, might narrow in space populations where the extremes proved maladaptive. Space humans might become more uniform in their social temperaments, or at least uniform in ways that permitted successful colony functioning. Alternatively, different colonies might develop radically different social configurations, selecting for different personality profiles. Some colonies might embrace fully communal living that rewarded highly social, highly affiliative personalities while making solitary types miserable. Others might develop strong norms of personal space, privacy, and social distance that favored more introverted types, while overwhelming those requiring constant social contact. The diversity of possible colonial social systems would create corresponding diversity in the personalities those systems selected. These personality shifts would not occur through any conscious decision or directed breeding program. They would simply emerge as certain types of people thrived and reproduced more successfully in certain environments while other types struggled. The social systems that colonies developed would reshape the psychological characteristics of their populations, which would in turn reshape the social systems those populations could maintain. Culture and psychology would co-evolve in feedback loops that might produce stable configurations unimaginable from Earth perspectives. Language evolution would proceed with remarkable speed in isolated space communities, creating communication barriers between populations within just a few centuries. Human languages change continuously even under stable conditions, with word meanings shifting, grammatical structures simplifying or complexifying, and pronunciation drifting across generations. Under normal conditions, these changes occur slowly enough that adjacent generations can communicate without difficulty. Grandparents and grandchildren share a mutually intelligible tongue, even if certain expressions or pronunciations seem old-fashioned or newfangled. Isolated populations experience dramatically accelerated linguistic drift because they lack contact with conservative language communities that might slow change through continued interaction. The English spoken in isolated Appalachian communities retained features lost centuries ago in British English because those communities had limited contact with the broader English-speaking world. Island populations developed distinctive dialects within just a few generations of settlement. Prison populations develop specialized vocabularies within years. Space colonies separated by travel times measured in years or decades would experience isolation-driven linguistic change, far exceeding any terrestrial parallel. Each colony would constitute its own linguistic island, free to drift in whatever direction its internal dynamics pushed without correction from outside communities. Even regular radio communication would slow but not prevent this drift, as the isolation would be social and practical even if not informational. Within a few centuries, different space colonies might develop mutually unintelligible languages, even if all began speaking the same mother tongue. The grammar, vocabulary, pronunciation, and pragmatic conventions would diverge independently in each colony, until speakers from different colonies could no more understand each other than modern English speakers understand Old English. They might recognize that they shared common linguistic ancestry without being able to communicate except through laborious translation. Technical vocabulary would diverge, as different colonies faced different challenges and invented different solutions requiring different terminology. The words for components of life support systems, for features of different habitat designs, for tools and techniques specific to different colonial circumstances would have no equivalents across colonies that had never needed those terms. Even basic vocabulary for features of daily life might diverge, as daily life itself took different forms in different colonies. Cultural concepts would lack translation between communities with radically different life experiences. How would a colony that had experienced catastrophic decompression events conceptualize safety compared to one that never had? How would a colony practicing communal child rearing discuss family, compared to one maintaining nuclear family structures. The categories through which people understood their world would differ so completely that mere word-for-word translation would fail to convey meaning. New concepts requiring new words would emerge in space environments that had no Earth equivalents. Earth languages lack vocabulary for three-dimensional navigation without gravitational reference, a central feature of microgravity life. They lack terms for the social relationships specific to confined space communities, relationships without terrestrial parallel. They lack words for psychological experiences unique to cosmos-dwelling populations, states of mind that Earth humans never encounter. Space languages would coin new terms, new metaphors, and new conceptual frameworks to describe realities unknown on Earth. These linguistic innovations would not merely add words to existing vocabularies. They would reshape the languages themselves, creating new grammatical structures, new patterns of expression, new ways of organizing thought through speech. After thousands of years, space languages might differ from Earth languages not just in vocabulary but in fundamental structure. The very architecture of thought might shift, as languages developed space-specific features that shaped how their speakers conceptualized reality. Linguistic research has demonstrated that languages genuinely influence cognition. Languages with different color terms affect how speakers perceive and remember colors. Languages with different spatial reference systems change how speakers think about space and direction. Languages with different temporal structures shape how speakers conceptualize time and causation. Space languages optimized for describing three-dimensional movement, artificial ecosystems, enclosed social worlds, and technological dependencies would shape the cognition of their speakers in ways that Earth languages could not achieve. After thousands of years of linguistic evolution, space humans might literally think differently than their Earth ancestors, because they would describe and categorize reality using different conceptual tools. Their inner mental lives might become inaccessible to Earth humans, not just through translation difficulty, but through fundamental cognitive incompatibility. Cultural practices would transform to serve the functional requirements of space living, while losing features that served only terrestrial functions. Human cultures evolved elaborate systems of ritual, art, religion, governance, and social organization over hundreds of thousands of years of terrestrial existence. These systems served adaptive functions in ancestral environments. Rituals promoted group cohesion and marked important transitions. Art transmitted values and created shared emotional experiences. Religion provided explanatory frameworks and moral guidance. Governance organized collective action and managed conflicts. These cultural systems would not simply transfer intact to space environments. They would face intense selective pressure from circumstances radically different from those in which they developed. The systems that survived would be those that served space-relevant functions. Others would fade, transform beyond recognition, or be actively abandoned as irrelevant to new realities. Rituals celebrating successful harvests would have no place in communities practicing hydroponic agriculture with continuous year-round production. But the human need for ritual, for marking achievements and transitions with ceremony, would not disappear. New rituals might emerge celebrating life support system maintenance milestones, successful expansion projects, the birth of generation-numbered descendants, or the anniversaries of founding voyages. The forms would change entirely while the functions persisted. Creation myths explaining humanity's relationship to the natural world would require complete reinterpretation in communities that had no natural world, that lived entirely within artificial environments of their own construction. The stories communities told about their origins might transform into accounts of the founding voyage, the struggles of early colonists, the heroes who solved critical problems that threatened colonial survival. These new origin narratives would provide the same psychological benefits as traditional creation myths, while addressing circumstances that traditional myths never imagined. Aesthetic traditions reflecting Earth landscapes, Earth creatures, and Earth phenomena might evolve into art forms celebrating artificial habitats, cosmic vistas, and the strange beauty of life in space. What would visual art look like in communities that had never seen a mountain, a river, or a cloud? What music would emerge from cultures that had never heard wind in trees or rain on roofs? What poetry would populations create who knew stars only as destinations rather than decorations? The aesthetic productions of space cultures might be as alien to Earth sensibilities as space environments themselves. Religious and spiritual beliefs would face particular adaptive pressures because many Earth religions involve concepts deeply tied to terrestrial experiences. Sacred lands that provide spiritual anchoring exist on Earth, not in space. Natural cycles that structure religious calendars depend on planetary orbits and rotations that space habitats do not share. Concepts of humanity's special place within creation often assume an Earth-centered cosmology that space living would directly contradict. Space religions might emerge addressing the existential concerns specific to space existence. Questions about humanity's purpose in the cosmos take on different urgency for populations actually living among the stars, rather than gazing at them from a planetary surface. The relationship between individuals and their artificial life support systems, upon which survival depends moment to moment, might acquire spiritual significance that nothing comparable existed to provide on Earth. The prospect of eternal expansion through space, of humanity spreading across the galaxy over millions of years, might inspire cosmic religious visions unimaginable to earthbound thinkers who could not escape their home world. Social organization would restructure around the practical necessities of space habitation rather than the historical accidents of terrestrial politics. Earth political systems evolved through competition and cooperation between groups of varying sizes in environments with abundant resources relative to population, mobility between political units, and the possibility of warfare as final arbiter of disputes. Space colonies would face entirely different structural constraints that might favor organizational forms unknown on Earth. Resource scarcity and the absolute dependency of all colony members on shared life support systems might promote more centralized control than typical of democratic Earth societies. When a single angry individual could potentially sabotage systems that everyone needed to survive, societies might develop mechanisms of social control more extensive than democratic traditions usually permit. The existential vulnerability of space habitation might override political values that assume survival is not constantly at stake. Alternatively, the technical complexity of space systems might favor distributed expertise and decentralized decision-making that empowered specialists over generalist leaders. No single authority could understand all the systems that colony survival required. Political power might flow toward those with critical technical knowledge, rather than those with traditional leadership qualities. Meritocracy of competence might replace hierarchies based on wealth, birth, or popularity. The impossibility of emigration would create intense pressure for effective conflict resolution mechanisms. On Earth, the ultimate resolution to intractable conflict was separation. Groups that could not coexist could move apart. Individuals who could not tolerate their societies could leave for others. Space colonies would offer no such escape. Every conflict would have to be resolved within the colony, because there was nowhere else to go. Societies might develop conflict resolution capabilities far exceeding anything Earth cultures achieved, because the alternative would be colonial collapse. Different colonies would likely develop different governance systems suited to their specific circumstances and founding populations. Some might evolve toward authoritarian technocracies, where engineering expertise determined political power and dissent was suppressed for collective survival. Others might develop radical democracies, where every decision required community consensus and individual rights were paramount regardless of efficiency costs. Still others might organize around extended family structures, religious hierarchies, corporate models, or arrangements with no Earth precedent. Over thousands of years, this diversity of colonial social experiments would reveal which organizational forms best promoted survival, reproduction, and flourishing under space conditions. Successful models would spread through cultural transmission to other colonies, and through colonial expansion as successful colonies founded daughter settlements. Failed models would disappear along with the colonies that adopted them, their organizational experiments terminated by extinction. Family structures would adapt to the reproductive requirements and social constraints of space communities in ways that might horrify or mystify Earth sensibilities. The nuclear family model that became dominant in industrialized Earth societies assumed conditions that would not exist in most space colonies. It assumed geographic mobility that allowed young adults to establish independent households away from parents. It assumed economic conditions that allowed single couples to support children independently. It assumed population sizes that provided ample mate choices without institutional coordination. Space colonies might lack all of these conditions. Alternative family arrangements from extended kinship networks, housing multiple generations under shared resource management, to communal child rearing that distributed parental responsibilities across entire communities might prove better suited to colonial demographics than Earth-style nuclear families. The family forms that emerged would be those that worked under space conditions, regardless of whether they matched terrestrial traditions. Small populations would face particular challenges around genetic diversity and mate selection that would force social innovations around reproduction. Earth cultural taboos against incest reflect the biological reality that inbreeding causes developmental problems through the expression of harmful recessive genes. In small space colonies, maintaining genetic diversity would require careful management that might conflict with individual preferences about reproduction. Different colonies might adopt different solutions to this challenge. Some might mandate reproductive pairings based on genetic analysis to maximize diversity regardless of individual romantic preferences. Others might embrace biotechnology to screen embryos for genetic problems, allowing more reproductive freedom while preventing the expression of harmful recessive combinations. Still others might actively promote immigration from other colonies specifically to refresh local gene pools with new genetic material. Each approach would shape family structures and reproductive norms in distinctive ways. Colonies practicing mandatory pairing might develop very different concepts of love, marriage, and family than those allowing free mate choice. Colonies relying on technological screening might develop different attitudes toward reproduction and children than those relying on social controls. The diversity of solutions would create diversity of cultures around the most fundamental human activities. Education would necessarily change to prepare children for lives radically different from anything Earth curricular anticipated. Earth education assumes certain background knowledge about the natural world that spaceborne children would never acquire through direct experience. It assumes future occupational structures that would not apply to colonial economies. It assumes cultural contexts that would not exist in populations for whom Earth was merely a distant ancestral home mentioned in history lessons. Technical skills related to life support, food production, habitat maintenance, and emergency response would likely become mandatory for all colonial citizens rather than specialized knowledge for particular professions. Every individual would need to understand the systems, keeping them alive well enough to respond appropriately to emergencies and contribute to maintenance. The generalist technical competence required for space survival would replace the specialized ignorance that Earth's division of labor permitted. Earth-centric, historical, and cultural knowledge would seem increasingly irrelevant across generations as lived experience diverged ever further from ancestral circumstances. Why would children who had never seen rain need to read poetry about it? Why would adolescents who would never visit Earth need to memorize its geography? Education would focus on knowledge that mattered for actual life, rather than maintaining connection to an ancestral home that grew more mythological with each passing generation. Identity itself would acquire new dimensions as old categories lost meaning and new ones emerged. Earth humans identify partly through national, ethnic, and cultural affiliations tied to terrestrial geography. Americans, Chinese, Nigerians, Brazilians. These categories would lose salience for populations who had never seen the territories those names referenced. New identity categories would emerge based on colonial origin, habitat type, generation number, technical specialization, or position within the expanding human presence in space. The fundamental question of what makes someone human might receive different answers from different space communities. Biological criteria would become problematic, As populations accumulated different adaptations, would beings with fundamentally restructured skeletons and different cardiovascular systems still count as human? Cultural criteria would fragment as incompatible traditions diverged beyond mutual comprehension. Philosophical frameworks for human identity would need to accommodate diversity that Earth humanity had never faced. After 10,000 years, the psychological and cultural transformation of space humanity might prove even more profound than the biological changes the previous sections described. Bodies adapt to environments through the slow mathematics of genetics and selection. Minds create meanings through the faster processes of learning, culture, and deliberate choice. The meanings space humans would create, the thoughts they would think, the societies they would build, and the purposes they would pursue might diverge so far from Earth patterns as to become genuinely alien while remaining recognizably human in some deeper sense that would itself require definition. This psychological and cultural divergence would combine with biological divergence to create something unprecedented in human history. Not just different populations of the same species as human groups have always been, but potentially different kinds of beings whose kinship derived from shared ancestry rather than shared present reality. The implications of this branching would reshape what humanity means and what futures remain possible. 10,000 years ago, modern humans were spreading across the globe in one of the most remarkable dispersals in the history of life on Earth. Small bands ventured into territories no human had ever seen, adapting to environments ranging from Arctic tundra where temperatures plunged far below freezing to tropical rainforests where humidity never relented. They crossed mountain ranges, navigated coastlines, built boats to reach islands visible on distant horizons. Within a few thousand years, humans occupied every continent except Antarctica and had begun adapting to local conditions in ways that would produce the diversity of human populations we observe today. Yet throughout this extraordinary expansion, these populations remained connected. Occasional migrations carried genes and innovations across the expanding human range. Trade networks linked distant communities. Intermarriage occurred wherever groups encountered each other. The genetic and cultural unity of humanity persisted despite geographic separation, because that separation was never absolute. Land routes and coastal voyages maintained threads of connection that prevented complete isolation. 10,000 years from now, humanity dispersed across space would face a fundamentally different pattern, with fundamentally different consequences. Instead of spreading across a single planetary surface where all points remained theoretically reachable, space populations would scatter across the three-dimensional volume of the cosmos. Instead of remaining connected through journeys measured in weeks or months, colonies would be separated by voids requiring years or decades to traverse, even at velocities approaching significant fractions of light speed. The conditions for speciation, the evolutionary process that creates new species from old, would be present as never before in human history. The biological species concept that scientists use to categorize life defines species as populations that can interbreed and produce fertile offspring. By this definition, all living humans belong unambiguously to a single species. Any human can theoretically reproduce with any other human, regardless of ancestral population, geographic origin, or physical appearance. The genetic differences between the most divergent human groups remain far too small to prevent successful reproduction. We are one species despite our superficial diversity. Space dispersal threatens this reproductive unity in ways that terrestrial dispersal never did. As isolated populations accumulate different genetic changes across hundreds of generations, their biological compatibility might gradually decline through multiple mechanisms that evolutionary biology has documented in other species undergoing similar divergence. Chromosomal rearrangements represent one pathway toward reproductive isolation. Chromosomes occasionally undergo inversions, translocations, and other structural changes that do not necessarily harm individuals carrying them, but can cause problems when those individuals attempt to reproduce with individuals carrying different chromosomal arrangements. If one population accumulates a chromosomal inversion, while an isolated population does not, offspring from matings between the populations might experience reduced fertility or increased rates of developmental abnormality. Accumulate enough such differences, and reproduction between populations becomes impossible. Developmental timing differences could create additional barriers. The precise choreography of embryonic development depends on genes being activated in specific sequences at specific times. If isolated populations evolved different developmental timing as they adapted to different environmental conditions, hybrid embryos might experience fatal timing mismatches. Genes inherited from one parent might activate too early or too late relative to genes inherited from the other, disrupting the delicate coordination that successful development requires. Immune incompatibilities could cause maternal rejection of genetically distant fetuses. The human immune system already walks a fine line during pregnancy, tolerating the foreign genetic material of the fetus while remaining vigilant against genuine threats. This tolerance depends on specific molecular signals that evolved within a context of reproductive compatibility. If populations diverged enough that these signaling systems no longer matched, maternal immune systems might attack fetuses conceived through interpopulation mating, causing miscarriage or developmental damage. These barriers would not appear suddenly like walls erected overnight. They would build gradually as populations diverged, each generation slightly less compatible with distant cousins than the generation before. The first interpopulation matings after centuries of separation might produce offspring with barely detectable problems: a slightly elevated miscarriage rate, marginally reduced fertility among hybrids. Problems that statistical analysis might detect but that would not prevent reproduction entirely. As divergence continued, these problems would compound. Hybrid fertility might decline from 90% of normal to 80% to 50% to near zero. At some point, a threshold would be crossed where reproduction between populations became so difficult that it effectively ceased even when individuals from different populations encountered each other. The populations would have become separate species by any biological definition, no longer sharing a common gene pool, no longer capable of merging back into a single reproductive community. The process might take far longer than 10,000 years to produce complete reproductive isolation. Speciation typically requires hundreds of thousands or millions of years in terrestrial contexts, but the trajectory would become unmistakable long before the endpoint arrived. Biologists studying human populations after 10,000 years of space dispersal might already observe the warning signs. Reduced hybrid fertility, increased developmental abnormalities in mixed origin offspring, the statistical signatures of incipient speciation appearing in demographic data. Even without biological barriers, social and cultural divergence might effectively isolate populations from one another as thoroughly as any genetic incompatibility. If space humans came to see inhabitants of other colonies as fundamentally different beings with incompatible values, incomprehensible customs, and alien appearances, social barriers might prevent interbreeding as effectively as biological ones. Species can exist as much in minds as in genes. The decision not to reproduce with someone is as effective at preventing gene flow as the inability to do so. Cultural speciation of this sort has precedence on Earth. Human populations have sometimes maintained separation for centuries despite geographic proximity and biological compatibility. Religious prohibitions, caste systems, ethnic prejudices, and legal restrictions prevented intermarriage between groups that could have easily interbred. These social barriers eventually broke down in most cases as cultures changed and intermixed. But space populations might maintain separation indefinitely if physical distance reinforced cultural difference and neither side saw reason to bridge the gap. The relationship between Earth-based and space-based humanity would grow increasingly strained as divergence accumulated across generations. The first generations of space colonists would maintain strong psychological ties to Earth, communicating regularly with friends and family left behind, following Earth news and cultural productions, identifying primarily as terrestrial humans who happened to live temporarily away from home. They would celebrate Earth holidays. They would teach their children Earth languages and Earth history. They would think of themselves as expatriates rather than as members of a new civilization. Subsequent generations born in space would feel progressively less connection to a planet they had never experienced except through stories and screens. Earth would become increasingly abstract to them, a place their grandparents came from rather than a home they might someday return to. The emotional resonance of Earth imagery would fade as lived experience diverged ever further from anything Earth could provide. Children born in space would find Earth landscapes strange rather than nostalgic, Earth customs quaint rather than familiar. By the 100th generation, roughly 2,500 years into the dispersal, Earth might seem as mythological to spaceborne humans as the Garden of Eden seemed to medieval Europeans, a place of origin featured in stories but bearing little relevance to present life, a symbolic homeland rather than a real destination. Distant ancestors came from there, the stories would say, but those ancestors had become legendary figures rather than relatable people. The connection would be historical rather than personal. By the 300th generation, roughly 7,500 years into the dispersal, even the stories might fade into fragments of fragments. Earth would become one origin myth among others, competing with accounts of the founding voyage, the early colony struggles, the heroes who saved populations from various catastrophes. The emotional weight that Earth carried for early generations would dissipate entirely. Earth humans would become strange others, as alien to space descendants as space descendants would seem to them. This estrangement would cut both ways with equal force. Earth populations would watch their space cousins become increasingly unfamiliar through whatever communications continued across the void. Bodies that looked wrong, proportioned differently, moving in ways that seemed unnatural. Languages that sounded bizarre, filled with unfamiliar words and strange rhythms. Customs that seemed incomprehensible, organized around concerns that made no sense to planet dwellers. The human family would become a collection of strangers who shared ancient ancestors but little else. Parents sometimes struggle to understand children who grow up in different circumstances than they did. Grandparents often find grandchildren's worlds bewildering. This generational gap, multiplied across hundreds of generations and combined with radical environmental divergence, would produce mutual incomprehension far exceeding anything terrestrial humanity has experienced. Earth humans and space humans might eventually struggle to recognize each other as kin at all, seeing instead beings whose resemblance to themselves seemed superficial and possibly coincidental. Communications between Earth and distant colonies would become increasingly difficult and decreasingly meaningful, even if the technological capability persisted. The time delays imposed by light speed limits would make real conversation impossible across interstellar distances. A message sent to a colony 10 light-years away would arrive 10 years after transmission. The response would arrive 10 years after that. A simple exchange of greetings would span 2 decades. Anything resembling dialogue would be impossible. Communication would become more like correspondence between historical archives than dialogue between living communities. Messages sent would describe conditions that had changed by the time they arrived. Responses would address concerns that had evolved before they could be received. Each party would be communicating with the past of the other, never the present, never able to catch up to simultaneity. The content of communications would become increasingly difficult to translate as languages diverged and conceptual frameworks separated. Earth humans would struggle to understand messages from beings who thought in 3-dimensional space without gravitational orientation, who lived in artificial ecosystems of their own construction, who organized their societies around principles having no Earth equivalents. Space humans would find Earth communications equally baffling, filled with references to weather and seasons, to nations and territories, to concerns utterly foreign to their experience of existence. Technical translation might handle vocabulary differences. If space humans invented a word for some component of their life support systems, Earth translators could learn what the word meant and render it in Earth languages. But the deeper conceptual gaps would resist translation entirely. How would Earth humans understand the social relationships that space humans had invented to manage their unique circumstances? How would space humans understand references to experiences they had never had and could not imagine? Trade between populations would face similar barriers that would erode whatever mutual benefit might otherwise motivate continued contact. The vast distances of space make physical exchange impractical, except between nearby communities. The energy cost of accelerating mass to interstellar velocities and then decelerating it at the destination would exceed the value of almost any cargo. Only information would be economical to transmit across such distances. Information trade would persist longer than physical trade, but even information would lose value as circumstances diverged. Technologies developed for terrestrial conditions might prove irrelevant to space needs. Agricultural techniques for Earth crops would not help colonies growing entirely different food sources. Medical treatments for Earth diseases would not address health problems unique to space environments. Cultural products created for Earth audiences might not translate to space sensibilities at all. Each branch of humanity would increasingly turn inward, developing according to its own circumstances rather than maintaining connection to the broader human family. The unity that characterized terrestrial humanity throughout its history, however fractured by conflict and competition, would give way to genuine separation. Multiple humanities would emerge from the single human trunk like branches growing from a tree, each heading in its own direction, each responding to its own light. Technology might either accelerate or retard this branching process, depending on how it developed and how communities chose to use it. Advanced communication systems might maintain connections across greater distances and timescales than baseline human psychology could manage. Artificial intelligences might translate between diverging languages and conceptual frameworks. Virtual reality might provide shared experiences that physical separation would otherwise prevent. Technology might serve as a bridge that evolution and culture would otherwise burn. Genetic engineering might correct accumulating incompatibilities before they became severe enough to prevent reproduction between populations. If molecular biologists could identify the chromosomal rearrangements, developmental timing shifts, and immune incompatibilities that threatened reproductive isolation, they might be able to engineer corrections that restored compatibility. Populations that would otherwise have become separate species might remain interfertile through technological intervention. Rapid transportation technologies might enable physical contact between populations that would otherwise never meet. If vessels could cross interstellar distances in years rather than decades or centuries, the isolation that drives speciation would be reduced. People might actually travel between colonies, carrying genes and cultural innovations with them. The threads of connection that prevented speciation during terrestrial dispersal might be maintained artificially across cosmic distances. Technology might preserve human unity that evolutionary forces would otherwise dissolve. But this preservation would require deliberate choice, continuous investment, and coordination across communities that might not share the goal. Some populations might value connection to the broader human family. Others might prefer their independence. The technological capability to maintain unity would not guarantee the social will to use it. Alternatively, technology might enable divergence far faster than natural selection alone could achieve, actively accelerating the branching rather than retarding it. Genetic engineering could adapt populations to specific environments far more quickly than evolution's glacial pace. Rather than waiting thousands of years for selection to favor bone density variants suited to microgravity, engineers might modify the genome directly to produce the desired traits within a single generation. Cognitive enhancement could create mental capabilities that differed radically between populations that adopted different modification strategies. One colony might enhance mathematical reasoning, another might optimize for social intelligence, a third might expand memory capacity, a fourth might accelerate processing speed. Each enhancement package would produce minds that excelled in different ways and potentially thought in incompatible patterns. Technology might not merely adapt humans to space, but transform them into beings that natural evolution could never have produced given any amount of time. Genetic modifications might incorporate capabilities from other species or invented from scratch. Cybernetic enhancements might merge biological and technological systems into hybrids that transcended traditional categories. The humans of the far future might be engineered rather than evolved, designed rather than selected, created according to plans rather than shaped by accident. The prospect of deliberately directed human evolution raises ethical questions that space communities would need to address without any guidance from Earth traditions that never contemplated such possibilities. What modifications are acceptable? Who decides what counts as improvement versus degradation? What happens to those who reject modification in populations where modification becomes normal? How do modified and unmodified humans relate to each other? These questions have no clear answers because they depend on values that would themselves diverge across separated populations. What one community considers beneficial enhancement, another might consider horrifying mutilation. What one community views as preserving human dignity, another might see as stubborn refusal to adapt to reality. The ethical frameworks for evaluating human modification would themselves evolve differently in different communities, making cross-community moral judgments nearly impossible. Some colonies might embrace radical modification, seeking to optimize their populations for their specific environments regardless of how far the results diverge from ancestral human form. If survival required beings with radiation-resistant DNA, why not engineer that resistance directly? If their environment favored beings with 4 arms rather than 2, why not add the extra limbs? If their circumstances rewarded minds structured differently from Earth-evolved brains, why not restructure the brain? For such communities, the ancestral human form would be a starting point rather than a sacred template. Other colonies might reject modification entirely, viewing their terrestrial inheritance as sacred and worth preserving, even if it makes space life more difficult. Such communities might maintain Earth-like bodies through pharmaceutical and mechanical support rather than through genetic alteration. They might accept higher mortality rates as the cost of preserving what they would see as authentic humanity. For them, becoming something other than human to survive would not constitute survival in any meaningful sense. Most colonies would probably fall somewhere between these extremes, accepting some modifications while rejecting others according to local values and circumstances. Minor adaptations that improved health without altering appearance might be widely accepted. Modifications that changed how people looked or thought might be more controversial. Each community would draw its lines differently based on its own ethical development. The result would be even greater diversity than natural selection alone would produce. Not just multiple human populations adapted to different space environments through evolutionary processes, but multiple types of enhanced or modified beings shaped by deliberate technological intervention. Some colonies might contain beings barely recognizable as human by ancestral standards. Others might contain beings almost indistinguishable from their Earth ancestors. The spectrum between these extremes would fill with endless variety. The tree of humanity would branch and branch again, each fork representing a choice some population made about what to become. Some branches would pursue biological modification, others would pursue cybernetic integration, others would pursue cognitive enhancement, others would pursue deliberate primitivism. The forms of human existence that would emerge could not be catalogued in advance because they would depend on choices not yet made and technologies not yet invented. Yet for all this divergence, something might remain constant across every branch of the human tree. The human mind, however modified in its particulars, might retain certain fundamental capacities that defined humanness more essentially than any physical form. The ability to form relationships, to care about other beings, to experience love and loss. The capacity to communicate through language, to share thoughts and feelings, to create shared meaning through symbolic exchange. The drive to create meaning through culture, to build systems of value and significance that transcend individual existence. These capacities might constitute a deeper human nature that no amount of physical or cognitive modification could entirely eliminate without creating something that would no longer want to be called human at all. Even beings with forearms and engineered brains and lifespans of centuries might still love their children, still mourn their dead, still create art and tell stories and wonder about their place in the cosmos. The forms these activities took might be unrecognizable, but the underlying drives might persist. Space humans 10,000 years hence might look back at their Earth ancestors the way modern humans look back at ancient hominids, recognizing kinship while marveling at the distance traveled. They would see beings suited to a single small world, unable to survive without technological support in the environments their descendants found comfortable or even pleasant. They would recognize the seeds of their own capabilities in those ancestors, the curiosity and adaptability and social bonds that made space colonization possible. They would see how much those seeds had grown and transformed in cosmic soil. The philosophical implications of human branching would challenge conceptions of identity, meaning, and value that Earth humanity had developed over millennia of assuming human unity. If multiple distinct beings all descended from the same human ancestors but no longer shared biology, culture, or even cognitive frameworks, which of them would be the true heirs of human history? Would any of them have better claim than others to the legacy of Shakespeare and Einstein and Buddha? Would Earth humans view their space cousins as family, as strangers, or as something else entirely for which Earth languages had no word? Would they feel pride in what their descendants had become, or horror at how far those descendants had strayed from ancestral form? Would they see space humanity as fulfillment of human potential or betrayal of human nature? Would space humans see Earth humans as honored ancestors whose memory deserved preservation, or as evolutionary dead ends who had refused to adapt when adaptation became necessary? Would they feel gratitude toward the beings who launched them into space, or would Earth seem as irrelevant to them as the African savanna seems to modern city dwellers? Would they consider themselves human at all, or would that term become archaic, referring only to the ancestral species from which they had emerged? These questions would have no definitive answers because the concepts underlying them would themselves be products of particular evolutionary and cultural trajectories. What counts as human, what constitutes identity, what makes life meaningful, are not fixed truths embedded in the structure of reality. They are variable constructions that different communities build differently based on their experiences and needs. Different branches of the human family would construct these concepts differently, and none would have authority to declare their construction correct. The cosmic perspective suggests that this branching might be not only inevitable, but desirable when viewed from a sufficiently expansive vantage point. A single species confined to a single planet faces extinction from any sufficiently large disaster, an asteroid impact, a nearby supernova, a pandemic of unprecedented lethality, a self-inflicted catastrophe of sufficient magnitude. Any of these could end humanity entirely if humanity remained concentrated in one location. A species spread across multiple worlds in multiple forms achieves a kind of collective immortality that no single population could attain. No single catastrophe could destroy all branches of a diversified human family. Some colonies might perish while others survived. Some lineages might go extinct while others flourished. The persistence of humanity as a whole would no longer depend on the survival of any particular population. From this perspective, the transformation of humanity over 10,000 years in space would not represent loss of human identity but its fulfillment. The capacity to adapt, to explore, to become something new in response to new challenges was always part of what made humans distinctive among Earth's species. Humans transformed every environment they entered and transformed themselves in the process. Space would simply provide new territories for this ancient tendency to express itself at scales previously impossible. Earth humanity domesticated plants and animals, transforming wild species into forms suited to human purposes. Wheat and rice and corn bear little resemblance to their wild ancestors. Dogs and cattle and chickens have been reshaped by thousands of years of selective breeding. Humans remade Earth's biosphere according to human needs and preferences, creating a world that could never have existed without human intervention. Space humanity would domesticate itself, transforming the human form into varieties suited to cosmic existence. The same creativity that reshaped Earth's plants and animals would reshape humanity itself. The same adaptability that allowed humans to thrive from Arctic to tropics would allow human descendants to thrive from orbital habitats to planetary surfaces to environments we cannot currently imagine. Humanity would become its own greatest domestication project. What would emerge could not be predicted in detail from our present vantage point. Evolution responds to conditions that cannot be fully anticipated. Cultural change follows trajectories shaped by countless individual choices and chance events. Technology develops according to discoveries that cannot be planned. The future branches in directions that no one standing at the trunk can foresee, but the general pattern seems clear enough. Dispersal into space would trigger the most dramatic transformation in human history, breaking the biological and cultural unity that had characterized our species throughout its terrestrial existence. The humans, who might someday look back at Earth from across 10,000 years of cosmic adaptation, would be our descendants in the genealogical sense, carrying genes that originated in Africa and passed through every generation between then and now. But they would not be us in any sense that present-day humans would find comforting. They would carry genes modified by millennia of natural selection in environments we can barely imagine, further modified perhaps by deliberate engineering toward goals we cannot anticipate. They would speak languages descended from those spoken today but evolved into forms we could not understand even with years of study. They would organize their societies according to principles developed to solve problems we have not yet encountered. Principles that might seem as alien to us as our democratic capitalism might seem to ancient hunter-gatherers. They would be the children of Earth who became the children of space. They would be the descendants of terrestrial primates who became cosmic beings. They would be humans who became something more than human, while remaining human in some essential sense that would persist across all the changes, some continuity of curiosity and caring and creativity that connected them to ancestors they could barely imagine. 10,000 years is both an enormous span and a small step when measured against different scales. Measured against a human lifetime, it stretches beyond imagination, beyond any ability to conceive what living so long would feel like. Measured against evolutionary time, it represents a brief interval, less than 1% of the time since humans and chimpanzees shared a common ancestor. The changes that would occur in 10,000 years of space habitation would be just the beginning of a transformation that might continue for millions of years if humanity survives and spreads. The divergence visible after 400 generations would be a preview of divergence that could eventually produce descendants as different from present humans as humans are from fish. The branching that would begin in our children's children's children would eventually produce a family tree as varied as all of life on Earth. What begins with bone loss and circadian disruption among the first astronauts would extend through the emergence of new species, new cultures, new forms of intelligence and existence. The cosmos would become a laboratory for human potential, exploring possibilities that terrestrial existence never permitted because terrestrial environments never demanded them. The question of what humanity might become would receive answers beyond anything we can currently imagine, answers written in flesh and culture and technology across distances we can barely measure. And somewhere, perhaps, beings descended from humans would look up at unfamiliar stars and wonder about their origins, just as we look up at our stars and wonder about ours. They would piece together the story of their ancestors from fragmentary records, genetic evidence, and archaeological remains scattered across worlds we have not yet visited. They would try to understand how creatures adapted to a single small world ever found the courage to venture into the cosmic darkness that those creatures found so terrifying and so beautiful. They would be us, transformed beyond recognition, and we would be them in potentia, containing already the seeds of everything they would become. The thread connecting present to future would remain unbroken even as everything about its form changed beyond any present ability to foresee. That continuity amid transformation is perhaps the deepest truth about what 10,000 years in space would do to humanity. It would make us more than we are while keeping us what we always were at the deepest level. It would make us what we always had the potential to become, potential that Earth alone could never have revealed. It would scatter the seeds of Earth across the cosmos and watch them grow into forms as various as all the environments the cosmos contains. The universe would take what Earth created and make it its own, and what it made would look back across the ages with wonder at the small blue world where it all began.